So conversation last time, we stopped at I believe somewhere here. Is that right? So just as a quick reminder, what we were talking about, we figured out that there are three parts of the ear: middle, external, and internal, not in order, not in the proper order. That auricle focuses the sound into the external acoustic meatus. External acoustic meatus transmits the sound to the tympanic membrane, which vibrates and transmits the sound to the bones called auditory ossicles. And it's time to talk about those three bones properly. Let me see if I do have them. If somebody can help me and grab those guys. Okay, just take them and run them just to take a glance. It's auditory ossicles. Look at them. Those are smallest bones. Pass them through the class. The smallest bones in your body. Okay? So they have you can name them both in a Greek and English fashion. So in the Greek fashion, it's going to be malleus, incus, and stapes. And we call them often the hammer, the anvil, and the stirrup. Okay? That's the order they exist. You know what fascinates me the most? You can see those bones, right? You can see they're noticeable. They're not like microscopic. You can see them with your naked eye. But what fascinates me the most is that these points where they are connected, these points right here, this is the synovial joint. So they are connected via synovial joints. We didn't talk about joints yet, but to give you an idea, this structure anatomically is similar to, say, your elbow or your knee. It's a proper joint, synovial joint with a cavity with a fluid in there. Pretty much, it's pretty neat. Okay. So the function of those bones is to vibrate and transmit those vibrations all the way to the oval window. Behind the oval window is the fluid called perilymph. We didn't talk about that fluid yet. We will. Perilymph. Now think about this. Can fluid transmit the sound? How do we detect the submarines? By echolocation, right? We can, special detectors can actually, quote unquote, hear the sound of the submarine from miles, tens of miles away. And before those detectors called sonars were designed, there were special people like sailors on the submarine who were sitting there listening to the submarines, or ships somewhere far. And we'll talk about the sound transmission and how it is transmitted in the different types of media. But the point is, fluid is a great sound transmitter. It's an excellent media to transmit the sound. So when those ossicles vibrate, those vibrations are transmitted to the fluid and they enter the realm of the inner ear. Does that make sense? So kind of, sort of, to repeat that, you have vibrations of the tympanic membrane. Those vibrations are transmitted to the auditory ossicles, and from auditory ossicles they are transmitted to the fluid via oval window. A couple of questions for you. First, each sound has a frequency, right? And each sound has a loudness. What happens to the frequency of the sound as it's being transmitted from tympanic membrane to the auditory ossicles to the fluid? Does it change or not? That's a common misperception. Loudness, yes. The amplitude changes, frequency doesn't. So the same pitch, say if you hear a bird singing, the bird sings at, I don't know, 441 hertz, you hear 441 hertz. The bird sings A of the first octave, you hear A of the first octave. Frequency doesn't change. The amplitude does. We will talk how it happens, but frequency doesn't change. Yes. So, the ossicles. So we start with malleus. Let me erase it, okay? We'll start with malleus, which is attached to tympanic membrane. Then it's incus, and then stapes. Bless you. That makes sense? Now, what happens when you go to the concert or to the nightclub and the music is really loud? What happens, like, initially? Huh? Well, yeah, I mean, you're pretty much deafened by the first experience, and then your sensitivity to the sound adjusts. Does that make sense? No. The part of that adjustment is mediated by the muscles called the tensor tympani and stapedius. Okay? Tensor tympani and stapedius are little tiny muscles. Stapedius is attached to the stapes. It is not shown here in the picture. And tensor tympani is attached to the malleus. These muscles, stapedius and tensor tympani, limit the vibrations of the auditory ossicles. Does that make sense? Think about this. If you hear really large sound, what's going to happen to the tympanic membrane amplitude? The vibrations will be really, noticeable and profound. Does that make sense? It will move, very much. Large amplitude, right? So those auditory ossicles will move with a huge amplitude as well. Does that make sense? They will vibrate vigorously. If they vibrate vigorously, it may damage them, and it may damage your inner ear because of those mechanical vibrations. Does that make sense? So you want to limit those vibrations. Muscles called stapedius and tensor tympani will contract in response to the loud sound and will limit those vibrations. Does it make sense? It's pretty much a negative feedback loop. Okay? We can picture it right here in the corner. So we have loud sound, which produce large amplitude of auditory ossicles. I presume you understand large amplitude like vibration amplitude, which leads to the muscle contraction, which leads to decreased amplitude of auditory ossicles. Does that make sense? You want me to repeat something here? Or decipher the chicken scratch? Is that understood? Okay. Now, we passed the... Listen up. If you kind of running behind and you want me to stay on the slide, just let me know. Don't hesitate, okay? So we passed the middle ear, and now we get into the structures of the inner ear, that is also called a labyrinth. I don't think I have it here, but we'll get to that when, we will work with the model. So this is your inner ear. Labyrinth has two parts, the bony labyrinth and the membranous labyrinth. So you have to appreciate that the bony labyrinth is kind of a cast. Does that make sense? Or it doesn't? Bony labyrinth is a cavity inside of the temporal bone. Does that make sense? It is a cavity. This cavity is filled with a fluid called perilymph. And the cavity, the bony labyrinth, consists of three parts: the cochlea, the vestibule, and the semicircular canals. Does it make sense? Now, a membranous labyrinth, which on the picture is shown as blue, okay, is a membranous structure, like tubular structure, that floats inside of the labyrinth, inside of the bony labyrinth. And in turn, that blue color, presumably, shows that the membranous labyrinth is filled with a fluid called endolymph. Okay? And endolymph has very high concentration of potassium. Membranous labyrinth consists of three parts. It's the cochlear duct. You can see it here, the blue one. It's utricle and saccule. It can be found in the vestibule, and semicircular ducts. Let me pause it for... So, again, you have three parts, and I'm going to show it to you on the model now. In terms of the bony labyrinth, you have cochlea, the spiral-shaped part. You have semicircular canals, and you have vestibule between the semicircular canals and the cochlea. Does that make sense? That's the bony labyrinth parts. This pink color, can you see the pink color? Represents perilymph. In that perilymph, the membranous labyrinth, that on the model it is shown as the gray structure, membranous labyrinth is suspended. Alright? Does that make sense? Membranous labyrinth consists of semicircular ducts, which you kind of can see, okay? Saccule and utricle. And you can't really see it, the cochlear duct. On this one, this cross section, the cochlear duct will be blue. You can barely see it, it's really tiny. Okay, that's cochlear duct. Does it make sense, more or less? It will make more and more sense as we go through that. Okay, so why fluid? Everything, both membranous labyrinth on the inside and bony labyrinth outside of the membranous, they are all filled with fluid, whether it's the endolymph inside of the membranous labyrinth or perilymph in the bony. Why fluid? Fluid is the great way to transmit the sound. Alright? That makes sense. Also, think about this. When you imagine that you hold a glass of water, and the level in that glass of water is even, and then you move to the side very fast. You shuffle to the side. What's going to happen to the water level? It's going to change, right? So that's why two parts of the inner ear, vestibule and semicircular canals, can sense the change of the, your position. When you change your position, when you move in space, that fluid that fills up semicircular ducts moves around. Does that make sense? As it moves around, this fluid movement is then transduced into the electric signals in the nervous system. So fluid allows you to do two things. First, hear the sound. Second, sense your position in space. Does that make sense? Inside of the cochlear duct, there is a structure called spiral organ. Spiral organ is responsible for the hearing sensations. In the saccule and utricle, there are structures called maculae. Maculae are responsible for sensing linear acceleration to the sides, or front and back, or up and down. And finally, crista ampullaris in the semicircular ducts, responsible for sensing rotational movements of the head. Those sensory organs, when they sense those mechanical changes in the inner ear, they transduce those changes into the electric impulses, nervous impulses, which they send to the central nervous system via vestibulocochlear nerve. Cochlear branch, you can see it here, is responsible for hearing, and vestibular branch is responsible for the balance sensing. So far, does that make sense to you? I'm not saying that, it's kind of, that's it, and you have to know it from now on. The point is, kind of reiterating it millionth time, two major chapters, the hearing chapter and balance chapter. Okay? And in both cases, if you think about that, balance involves movement. Sound involves movement. So all those structures sense movement. They detect movement. Okay? Good? Now, anatomy heavy part. Structure of the cochlea. As I mentioned before, cochlea is the spiral shaped part of the inner ear that you can see right here. Okay? It's the bony labyrinth, spiral bony labyrinth. Okay? Rather small. It says the size of the split pea. Well, you can only imagine how much this is bigger than the normal human ear. Well, it gives you pretty much the scale. Small. In the center of the cochlea, this bony structure is called modiolus. Like a pillar around which the spiral ladder goes. Okay? At the end, cochlea ends at so-called cochlear apex. This is cochlear apex right here. Okay, apex generally in anatomy means top, upper portion. Inside of the cochlea you can see three ducts. Can you see them? Duct, duct, and duct. Can you appreciate those guys? Can you see that? Three ducts. They run pretty much in parallel. Sorry. This is cochlear duct, or scala media. Inside of the cochlear duct, you will find so-called organ of Cordy. Okay? that organ of chordia, spiral organ, is the structure immediately responsible for sensing the sound, for transducing the mechanical vibrations, which sound is, into the nerve impulses. Okay? Does that make sense? Now, this, together with osseous spiral lamina, which is the extension of the bone, cochlear duct and osseous spiral lamina divide the cavity into two more scales, scala vestibuli and scala tympani. Scala vestibuli and scala tympani are parts of the bony labyrinth. Okay? They are filled with perilymph, and they are continuous with each other. Does that make sense? Let's talk about it one more time. So you have several scalae. You have cochlear duct, which is the part of the membranous labyrinth. Okay. You have scala tympani, and you have scala vestibuli. Now you have to appreciate that this is the cross section. However, if you would look at this model, if you have some level of imagination, you can imagine that these scala are running through the cochlea, okay, and they end up at the cochlear apex. Side of the apex, there is a little structure called helicotrema. At helicotrema, scala vestibuli becomes scala tympani. Think about this. It's like a road. You know, you drive down the road. Listen, you drive down the road, and at the very end of the road, it's the dead end. And all you can do is to turn around and go the other way, back. Does it make sense? That's what happens with the sound, actually. It goes through scala vestibuli, in some cases turns around, and goes back through the scala tympani. Okay? That makes sense? Both scala vestibuli and scala tympani are filled with perilymph, so they are parts of the bony labyrinth. Scala media, or cochlear duct, is filled with endolymph, and it's a part of the membranous labyrinth. Okay? These ducts are separated by the several membranes. Scala vestibuli and scala media, or cochlear duct, separated by the vestibular membrane. Scala tympani and scala media are separated by the basilar membrane. Okay? Stria vascularis, which can be found in the wall of the scala media, it produces endolymph. Okay? And finally, the cochlear nerve approaches the spiral organ, and essentially the function of it is to transmit the electric impulses, the nervous impulses corresponding to the sound, to the central nervous system. Okay, let's keep going. And we got the anatomy, okay? We have three scala. Scala vestibuli, scala tympani are continuous with each other at helicotrema, filled with perilymph. Scala media is in between them, filled with endolymph, and that's where the organ of chordia is. Now we're going to talk about fun stuff, physics. The sound. Sound is a mechanical wave. Does that make sense? It's pretty much a wave that is composed of the areas of compression and rarefaction. Areas of the higher medium density and lower medium density. Sound can be produced by practically any vibrating object. Okay, the only thing that it requires is the medium that consists of a matter. Sound cannot be transmitted in vacuum. So next time when you watch Star Wars or Star Trek or whatever and hear explosions in space and think it cannot happen, thank me for ruining your movie experience. Sound can be heard in the air. Sound can be heard in the water. Sound can be heard in the ground. You may have seen in the old Western movies how, these trappers, they put their ear on the ground and they say, the bandits are like 40 miles away, I hear the horses. Maybe not 40 miles, but you definitely can hear that. In fact, sound spreads to the farther distances in the water or ground than it does in the air, because water and the solid medium is denser. Does that make sense? Okay. Now, sound can be characterized by frequency or the wavelength. Those parameters are opposite to each other. Okay. High frequency corresponds to the short wavelength. Look at this picture here. The red wave has the higher frequency. The blue wave has the lower frequency. However, in a given moment, you have two red waves and one blue, which means blue wave has the longer wavelength. Does that make sense? So they are not the opposite, they are inversely related. The higher the frequency, the lower the wavelength. Does it make sense? Frequency corresponds to the pitch of sound. The higher the frequency, the higher the pitch. One frequency that I remember is the frequency of the A, the first octave. If any one of you plays musical instrument like piano or any other, that A sound that is used to tune up the orchestra, the very first sound, if you go to the concert, the very first sound, I think it's oboe that makes this sound. It's A of the first octave, it's 441 hertz. 1 hertz corresponds to 1 wave per second. So frequency of the blue wave here will be 100 hertz because it's 1 wave in 0.01 second. The frequency of the red wave is 200 hertz. This is 2 waves per 0.01 seconds. Does that make sense? Higher, say again? Higher frequency is the lower wavelength, smaller waves. Human ear can sense the sounds between 20 hertz and 20,000 hertz. But the sounds that we hear the best are in between 15 and 4,000 hertz. Okay? Does that make sense? At this frequency, we can distinguish sounds that are different in terms of the frequency by 2-3 hertz. It is very little. It's less than a half tone for those of you music aficionados. We can tell apart sounds that are different by half tone. We all have very good hearing. Does that make sense? Sounds that have frequency lower than 20 are called infrasounds. Sounds that have frequency higher than 20,000 are ultrasounds. At about 15,000 hertz, we can barely hear it. Really, it's really hard to hear. Dogs can hear it very well. And that's one of the mechanisms, you know, this dog repellents, sound dog repellents. Dogs can hear that sound at 15,000 hertz very well. Since I'm a runner, I know what I'm talking about. You press the button every time the dog barks, generates that sound. Dogs feel it as a very unpleasant sound, and it forms the, you know, negative reinforcement for barking. Do you understand what frequency is? Amplitude is the loudness. It corresponds to the energy of the sound wave. So Mari, you were right, energy changes, but it changes the loudness. Frequency, as we perceive it, doesn't change. Okay? So essentially, loudness is the height of the sound wave. The blue wave here is louder than green wave, right? Loudness is measured in decibel. This is a little bit tricky unit. First of all, we can measure the energy of the sound, or I would say, you know, intensity of the sound, as the power per surface unit, watts per square meter. The higher the intensity, the louder the sound. Now, it's really hard to operate in these units. They aren't perceived very relevant. Do you know what I'm trying to say? Like if I tell you, sound has 137 watts per square meter. What does that can mean? Okay, I don't know what it is. Okay, but we use decibel. Now, I want to look in the eyes of the person who created that unit, because it's totally counterintuitive. Not totally counterintuitive, but somewhat. Each 10 decibel correspond to increase in amplitude by tenfold. Let me explain it in the normal words. We go 10 decibel more, the sound becomes 10 times louder. We go 20 decibel more, the sound becomes, listen closely, 100 times louder. We go 30 decibel more, sound becomes 1,000 times louder. Do you see the pattern? Every time you add 10 decibel, you have to multiply the loudness by 10. Does that make sense? It's kind of a weird way. So please understand the difference between the frequency / wavelength and amplitude. Which sound characteristic do they correspond to? Does that make sense? Awesome. The fun stuff. We have really good hearing. If you would look at this representation, those are frequencies that humans and other animals can hear. Whales and dolphins are better. Other than that, we're pretty good. We hold probably like second place. See? We can hear very low frequency sounds. Sorry, very high frequency and very low frequency, because it's not a frequency, wavelength. Okay, the short wavelength means high frequency. The long wavelength means low frequency. Whales and dolphins, that's the only way to communicate, produce sounds underwater. Okay, we're better than rodents. We're better than birds, fish. Well, you got to be better than fish frogs, and so on and so forth. Some of the things are very interesting. Think about this. Bats, they can hear in the ultrasound region, right? Ultrasound part of the sound spectrum. Makes sense. They use sound to locate the objects. They produce sounds and then they sense that sound as it comes back from the objects. Okay. That kind of shows you the distribution of various sounds in terms of the frequency. So bass will be about 60 to 250 hertz, violin 250 to 2000 and a change. So you see, sounds that we can normally perceive, they all lie in the area from somewhat like 60 to 4 to 8,000. Does it make sense? Can we hear something lower than bass? Yes, of course. Not terribly well though. Can we hear extremely like high sounds? Yes, not terribly well. We may not be able to distinguish very well between different pitches. Does that make sense? And finally, the interesting part, how loud different sounds are. I don't know, on the right you can see the scale, and it tells you the number of times, threshold of hearing, okay? Normal breathing is 10 times louder than the threshold of hearing. Rustling leaves, 20 decibel, 100 times louder. Empty theater, 30 decibel, 1,000 times louder. Can you see that relationship? Every time we add 10 decibel, we elevate the loudness tenfold. 10 times louder, 100 times louder, 1,000 times louder. Okay. Few things. That's the scale again for music aficionados, from pianissimo to fortissimo, the quietest to loudest. So, well, probably the loudest sound that one can produce playing in orchestra is the subway train, which is pretty loud. Now, a few things to notice. Threshold of pain is 120. Have you heard about the world record that was set at the Seahawks Stadium 2 or 3 years ago? Seahawks Stadium is considered to be the loudest stadium on earth due to the specifics of the bleachers design. In some areas of the stadium, the sound kind of is being focused and can reach extremely high numbers. The world record on Seahawks Stadium was set at 137 decibel, which is as loud as the jet engine. Okay. That's pretty impressive, right? Much higher than threshold of pain, so I bet all those people either went deaf or wearing the earplugs. Little factoid, I can tell you the loudest snore that is recorded in the Guinness World Records. It's 127 decibel. Huh? That is louder than the machine gun. It's the loudest, the man who has the loudest snore is surprisingly married. Not so surprisingly, his wife is one ear deaf. Huh? Well, probably so, yeah. Okay. In order to understand the physiology of the sound perception, We need first to figure out how sound travels through the ear. Okay? How does sound enter the ear? Through what? You start like in the middle. Yes, we start with an auricle, and auricle focuses sound waves into what? Auricle focuses the sound waves into external acoustic meatus, right? And then from external acoustic meatus, the waves reach, now they reach tympanic membrane. Does that make sense? So here in this picture you can see waves reaching tympanic membrane. What happens to the tympanic membrane when the sound waves reach it? It starts to vibrate. Does that make sense? It starts to vibrate. As it vibrates, those vibrations, where are they transmitted to? The auditory ossicles. Can you see them? So as they are transmitted to auditory ossicles, in which order they start to vibrate? First is malleus or hammer, second is incus or anvil, and the third is stapes or stirrup. Stirrup overlays the oval window. You can't really see it, it's under the stapes. Here you can see stapes. Okay? I don't know if we can. Oh, oval window right here. Okay? Stapes transmits the vibrations through the oval window into the scala vestibuli. Right here. And the fluid in scala vestibuli starts to vibrate as well. The vibrations on this picture are represented by this wavy arrow. Now, there are two options. One, vibrations are in the hearing range, between 20 and 20,000 hertz. Does that make sense? Another option, vibrations are still reaching the ear, they are out of the hearing range. They are either infrasound or ultrasound. The pathways are going to be different for two cases. If vibrations cannot be heard because they are out of the hearing range, here's what's going to happen. Look at this. The sound will go all the way to helicotrema through the scala vestibuli. Remember what I told you, scala vestibuli and scala tympani are continuous. So the sound will kind of circle around at helicotrema and go back through the scala tympani to the round window. Does it make sense? In case the sound is out of hearing range, it doesn't cross scala media. Can you see that? Option number two, sound is in the hearing range. In this case, sound will cross the scala media, sort of take a shortcut, all right? And this shortcut will cause the vibration of the basilar membrane. So think about this. If sound is in the hearing range, it will go through the vestibular membrane, scala media, into the scala tympani, and as it goes, it will vibrate the basilar membrane. that make sense? Basilar membrane has spiral organ, the hearing organ of Corti, right here. As basilar membrane vibrates, organ of Corti vibrates. And as it vibrates, it generates the electric impulses, and you've got the hearing sensation. We're going to talk about this part too. Do you understand the sequence? How sound goes through the ear. You can grab an ear. You have, some of you, do you have the ear? Jamie, you can grab the ear. I can do it without modal. Thank you. So it starts with the auricle. Okay, can you see the auricle? Auricle focuses the sound into the external acoustic meatus. From external acoustic meatus, sound vibrates tympanic membrane. Tympanic membrane transmits the vibrations on the auditory ossicles, malleus, incus, stapes. Stapes transmits vibrations through the oval window onto the perilymph in a scala vestibuli, and if the sound is in the hearing range, sound wave takes quote-unquote shortcut through the scala media into the scala tympani. As it takes the shortcut, it vibrates the basilar membrane, and then sound leaves through the round window, sort of dissipates through the round window. That makes sense? Oh, okay. Turns out I made a little animation, which is very unusual for me. So this is sort of the same stuff going in the form of a flowchart. So sound waves are generated, enter external acoustic meatus, set vibrations of the tympanic membrane at the same frequency. The louder the sound, the higher the amplitude. These vibrations are transmitted to the oval window by auditory ossicles. Now, a very important point here. Oval window, if you would look at your modals, take a glance at the stapes, the surface under the stapes, and the total surface of the tympanic membrane. Tympanic membrane is larger, about 20 times larger in surface than the oval window. So vibrations, all that energy of vibrations from the tympanic membrane will be transmitted to the much smaller oval window, which means amplitude will be increased about 20 times. So the difference in surface area between tympanic membrane and oval window contributes to the amplification of the sound. Okay? So vibrations through the oval window are transmitted to the cochlear fluid in the scala vestibuli, and then the sound is in the lower frequency. Vibrations go through scala vestibuli and scala tympani, never bothering scala media. If vibrations are in the normal frequency range, they will cut through the cochlear duct into the scala media, and basilar membrane will be activated, and the spiral organ will be activated. Does that make sense? So what is the function of the spiral organ? Spiral organ transforms the vibration into the nerve impulse. Structurally, it contains supporting cells, which, as the name suggests, support, and hair cells. You can see the hair cells on this micrograph. You can see the hair cells on this image, and you can see hair cells on this model. Okay. There is one row of the inner hair cells and 3 rows of outer hair cells. Now, if you would look at these hair cells, look at the model once again, you can see that the hair cells are between 2 membranes, the basilar membrane and the tectorial membrane. Okay? Look at the image again. That's the basilar membrane, and that's tectorial membrane. Hair cells are kind of stuck in between those two. Does that make sense? Only one of those membranes actually can move. It's basilar membrane. Tectorial membrane remains not moving. Okay. You can also appreciate that at the bottom of the hair cells there are fibers of the cochlear nerve. These fibers the afferent fibers, the sensory fibers that will transmit nerve signals to the CNS. Okay? Does that make sense so far? Why do we call hair cells hair? They have cilia on the apical surface. They have multiple cilia of different length, and they have the largest cilium that is called kinocilia. Okay? This structure, stereocilia and kinocilium, they extend in the endolymph. Remember, I told you before, look at this model once again. This is the scala media. It is filled with endolymph. So hairs, the cilia, are in the endolymph. Does that make sense? Stereocilia are linked together by the fibers called tip links. What it means, if my fingers are stereocilia, think about this. They're stereocilia. And if they are linked together, if one bends, others bend too. Does that make sense? If they all link together, one bends and others bend towards it too. Does that make sense? Okay? Now, the longest stereocilia are embedded into the tectorial membrane. You can't really see it on this model, but let me put it this way. I use this model very often. So my hand, this is basilar membrane. Okay? My palm is the hair cell. My fingers are stereocilia and kinocilium. My left hand is the tectorial membrane. Look at me and tell me if you can kind of understand what I'm trying to tell you. Stereocilia, my fingers are stereocilia. This is tectorial membrane. My forearm is basilar membrane. Can you refer this to the picture right there? Does that make sense? Let's take a glance what happens when sound crosses scala media. And sound vibrations across the scala media. What happens to the basilar membrane? Vibrations across the scala media. What happens to the basilar membrane? It moves. Basilar membrane, stereocilia, dictorial membrane. Basilar membrane starts to move. What happens to the stereocilia? They move too. Does that make sense? Here's what happens. These are stereocilia and kinocilia. Okay? As the basilar membrane moves, stereocilia deflect. They move towards kinocilium. As they move towards kinocilium, that tension, mechanical tension, opens potassium channels. And potassium flows into the cell. Does that sound weird, or does it sound normal? Potassium flows into the cell. Diffuses, actually. Does it sound weird? Does it? It does. Why? Usually potassium flows out. Usually potassium concentration inside of the cell is high, right? But in this case, which fluid surrounds hair cell? Endolymph. And endolymph has very high concentration of which ion? Potassium. So in this case, potassium concentration outside is very high, and potassium starts to flow into the cell. Does that make sense? What is the charge for potassium? Positive. When potassium flows into the cell, what happens to the membrane potential of the hair cell? Increases. Cell becomes depolarized. Depolarization leads to the additional opening of calcium channels. Calcium flows in. Calcium influx. What is released in response to the calcium influx? Neurotransmitter is released in response to calcium influx. Okay? Does that make sense? And then eventually, when sound is over, when there's no more sound, those, what you might call it, stereocilia bend backwards. Channels close. Repolarization occurs. Does it make sense? And the cell can transmit the signal once again. You got it? Right? Let's go over it again. Basilar membrane moves. Stereocilia bend. Potassium channels open. Since endolymph is potassium rich, potassium enters the cell. Cell depolarizes. Calcium channels open. Calcium flows into the cell. Leads to the release of neurotransmitter. Once the sound is over and stereocilia bend back, potassium channels close, cell repolarizes. If the sound is very loud, what's going to happen to, amplitude is huge, what's going to happen to the bending? Stereocilia, in response to loud sound, are they going to bend more or less? More. If they bend more, what about channel opening? You're going to be open for longer and maybe more channels. The more channels means higher potassium influx, means higher calcium influx, means more neurotransmitter released. More neurotransmitter, you have sound perception, louder. Does that make sense? I will ask you, about this process. I will ask you about the flow of ions. I will ask you about the neurotransmitter release. I will ask you about bending of stereocylin. Does that make sense? Questions? Couple of things. First, how can we tell apart the pitch? Turns out, basilar membrane has different fibers. You know, at the base, the fibers are shorter. At the land, the fibers are longer. It turns out the individual pitch will cause the vibrations of only specific fibers. If you ever played a string instrument, especially guitar, especially electric guitar, if you step with an electric, you take a note and you step back with an electric guitar to the amplifier, what's going to happen? Feedback. Why? Is it going to be all 6 strings? It's not going to be all 6 strings, because frequency of the sound that comes out of the amplifier will be able to vibrate one of the strings. Depending on the frequency, it's going to be different string. Same happens here. A particular pitch will vibrate a particular fiber. Does it make sense? So only hair cells located on that fiber will release glutamate. Okay? So only afferents that come from that fiber of the basilar membrane will send the signal to the brain. Does that make sense? Each individual pitch will vibrate one fiber. What if we have a chord or multiple pitches? Many people talk at the same time. The answer is simple. Yeah, more than one fiber will vibrate. Does that make sense? It's pretty mechanical. Do you understand that idea? Say again? Music? Oh. No, don't worry about that. No. What I'm trying to show, this picture shows you pretty nice. So high frequency, okay, shorter fibers being vibrated. Medium frequency, longer. Low frequency, the longest fibers vibrate. Okay. Is that understood? Now, we mentioned sort of en passe that there are 2 types of hair cells. The inner and outer. What's the difference? It turns out that afferent fibers that transmit the nerve signal to the brain, all those afferents are at the inner hair cells, and practically none of them at the outer hair cells. Why? Outer hair cells actually have a very interesting function. Think about this. If you hear a very loud sound, what can you tell about the sound wave amplitude? It is high. You got it? If the amplitude of sound is high, what can you tell about the amplitude of vibrations for basilar membrane? It's going to be what? High or low? High, right? Also high. If basilar membrane vibrates vigorously, how about the vibrations of stereocylin? Also high, right? Now think about this. There's more chance to cause mechanical damage to basilar membrane or to the cells if the amplitude is high. Does that make sense? Outer hair cells protect inner hair cells from damage. Here's how. If the sound is loud, okay, neurotransmitter is released from the fibers, efferent fibers, that innervate outer hair cells. Pretty much it's like a feedback. Sound is too loud. Signal to outer hair cells. Outer hair cells become stiff. Does it make sense? If these cells, you see this 3 rows right here? If these cells become stiff, they will limit the vibrations of basilar membrane. Look at this, right here. Outer hair cells will limit the vibrations of the basilar membrane. So they will protect inner hair cells from damage. Does that make sense? Now remember we were talking about, do you understand that idea? Loud sound, outer hair cells stiffen in response to that loud sound and limit the vibrations of the basilar membrane. So the inner hair cells do not vibrate like crazy, which essentially regulates the sensitivity of your hearing. That comes to the same point when we talked about the loud noises, loud music at the concert. When you first step in, okay, you're deafened by experience, right? But if you stay there for a while, you're okay. The only problem is that your sensitivity decreases so much when you step out. You cannot hear anything because your ears are not sensitive anymore. They those hair cells stiffened. Remember when we talked about the vestibulocochlear nerve? And we mentioned that it's 99% sensory. This is its motor function. In response to the loud sound, vestibulocochlear nerve sends the efferent signals to the outer hair cells in those 2 muscles in the inner ear, stapedius and tensor tympani, that limit the vibrations. That's very small motor role of the vestibulocochlear nerve. Other than that, it's sensory. Is that clear? So please understand the idea about different fibers of basilar membrane responding to different frequencies and the role of the outer hair cells in controlling vibrations of basilar membrane and protecting inner hair cells. Okay. We're going to finish audio and take a break afterwards. Okay. The route. So bipolar cells in the spiral ganglion receive the input from the spiral organ, okay, and then transmit that input to the cochlear nucleus. From cochlear nucleus, the auditory output goes via the midbrain, which is responsible for the auditory reflex. Remember the startle reflex? Okay. It goes to the thalamus, and what's important, some of those fibers decussate and some don't. Look at this. You have, so that's one ear, okay? Bipolar cells enter the cochlear nucleus, then through the medulla and midbrain, some of the fibers go to the other side, some of the fibers remain on the same side. Does it make sense? So it means that each hemisphere receives the auditory inputs from both ears. Does that make sense? Each hemisphere receives the auditory inputs from both ears. What I want you to know about the pathway. I don't care that much about the nuclei, but I want you to understand that it goes to the brain stem first, medulla and midbrain, thalamus second, and only afterwards to the auditory cortices. Okay? Now, quick review of how we perceive pitch, loudness, location of the sound. Pitch. What we just described. Different pitches will excite different sets of hair cells. Does that make sense here? Multiple pitches will be perceived, and each set of hair cells has its own little tiny afferent fiber. Think about this. If those are different fibers of the basilar membrane, the higher pitch will vibrate this fiber. Hair cells on this fiber will send the signal to the brain. That's one note on the keyboard. The lower frequency will vibrate this one. Hair cells on this one will send the signal to the brain, different note on the keyboard. Actually, if you think about it, there's, I don't know the name for that instrument, but in orchestra, when people play with a little, like, timpani. Yeah, so it's like little timpani, okay? You strike one plank, and it gives you one sound. So you strike one fiber, and it gives you one pitch. Does that make sense? Awesome. Loudness. Louder sound, higher amplitude, larger moves of the hair cells, higher graded potential, more neurotransmitter is released. That's what we were talking about. More neurotransmitter, more frequent action potential, greater loudness. Does that make sense? Last but not least, location of the sound. That's very interesting. It turns out we are amazing physiologically. Sound from, say, when I talk, James, his left ear is closer to me than his right ear. There's a little bit of difference in terms of timing. So sound of my voice reaches James's left inner ear first. Does that make sense? So there's a little bit of delay. Also, sound of my voice is a little louder in his left ear than in his right. Does that make sense? So difference in the loudness and timing as it reaches two ears allows us to tell where the sound is.
Talking to you face to face, would timing be equal?
Yes, but I cannot possibly imagine perfectly positioning myself. Does that make sense? So there still be, we're very sensitive to it. Okay. Now, pathology of the hearing. Two types of deafness and tinnitus. Conduction deafness, obstruction of the sound path. What can cause conduction deafness? Earwax, accumulation of earwax. You can consider, earplugs cause conduction deafness, right? When you put earplugs in your ear, sound just doesn't reach through. Does that make sense? Tympanic membrane, if it's destroyed or ruptured, it cannot effectively transmit the sound to auditory ossicles. Another problem, so-called otosclerosis. Those three auditory ossicles right here, they may fuse. Does that make sense? Imagine this. You have 3 separate bones. Well, I don't have anything here near me. You have 3 separate bones. And they vibrate because they have those little joints between them that allow them to vibrate. If joints are arthritic, essentially arthritis, joints fuse and you have like one bone that goes from oval window to tympanic membrane, okay? So in this case, they cannot transmit the sound. Sound is conducted by skull bones, which makes it like a little bit deaf and weird. Otosclerosis can be treated surgically. Those bones can be separated. Or if the bones fuse to the oval window, it can be separated. Does that make sense? So everything about the sound conduction, any problem in the conduction of the sound all the way to the oval window can cause conduction deafness. Sensorineural deafness. Damage to any types of nerves. For instance, hair cells. If somebody is exposed to extremely large sounds all the time and doesn't wear the protection, that vigorous vibration, and remember, outer hair cells, they can protect inner hair cells only so much. Vigorous vibrations will damage inner hair cells and you lose the hearing. Does that make sense? Any damage to the structures of the spiral organ. Okay? Hair cells die and, that's it. Older people usually have lower hearing just because of the lifetime exposure to the sound and, death of the hair cells and so on and so forth. Of course, damage to the nerve. Okay? Of any sorts. Tumor, stroke, the leading cause of the congenital hearing loss, congenital deafness in the United States is cytomegalovirus infection in newborns. Cytomegalovirus actually damages the nerve. It causes sensorineural deafness in newborns. Okay? Does that make sense? Bless you. There is a treatment called cochlear implant that can restore the hearing of those people. But that's the example of sensorineural deafness. Tinnitus, ringing or clicking. Essentially, it's the auditory hallucination. You hear the sound when there is no sound. I have tinnitus myself. From time to time, my ears start to ring. It's very, very weird. Okay? What can cause nerve degeneration? Sort of a symptom of the nerve degeneration. Otitis media, middle ear inflammation, can cause tinnitus. Some medications can cause tinnitus. Does that make sense? Any questions on the deafness and pathology and whatnot? You good? You understand the difference between conduction and sensorineural? Well, I'm going to